So you're welcome. In this class we'll be talking about pairwise alignment. We'll be working on pairwise alignment. And what is pairwise sequence alignment? So it's a method which are used to find the best matching pairwise, which is either local method or global method alignments of two query sequences. So that means we deal majorly with two sequences in pairwise alignment. We deal with two sequences, and most times these sequences can either be from different species or from the same species, but of different variations when it comes to mutation. So we use pairwise alignment to run mutational analysis, to embark on mutational research projects to understand the mutation that took place within two sequences. You understand in the mutant sequence, and understand the codons they were altered, the nucleotide bases that were altered, the amino acid they were altered. You understand now? So, and the effect of those alterations in the sequence when it comes to the functional in gene products, that is proteins. You understand? And so we're going to be dealing with that to understand what actually took place in certain organisms. Especially we'll be working on sickle cell. So what exactly took place in sickle cell, what alterations took place in sickle cell, what mutation took place in sickle cell, what are the effects of mutations in that in the hemoglobin beta gene that result to sickle cell, affecting hemoglobin and yielding to inability to transport oxygen to the throughout the body, supplying oxygens to other cells of the body. So we're going to be working on that so that we'll be able to understand what actually took place, and so that you can single-handedly handle mutation analysis when it comes to pairwise alignment method. Now pairwise alignment can also can only be used between two sequences at a time, but they are efficient to calculate and often used for methods that do not require extreme precision, such as searching a database for sequences with high similarity to a query. So searching a sequence database for sequences with high similarity to a query actually deals with multiple sequence alignment. So, and there are three ways, basic methods of working on pairwise alignment, and we have Dutch matrix method, which I already showed you, at DNA sequence alignment, DNA analysis, so, and then dynamic programming and Word's method. So I believe you will understand Word's method, especially when it comes to your theoretical knowledge in school. And this dynamic programming is actually the method we're going to embark on using GENEIOUS software. So just let me show you one thing about dot matrix. This is dot matrix, and here is telling us that dot matrix approach, which simply produce a family of elements for individual sequence regions. It's qualitative and conceptually simple, though time consuming to analyze on a large scale. In the absence of noise, it can be easy to visually identify certain sequences features such as insertions, deletion, repeats, and all of them. But this time around in this course, we'll be working on dynamic programming, which is used of GENEIOUS software. So it is now important for us to embark on GENEIOUS software to be able to work with pairwise alignment in understanding mutational research. So all you need to do is just go to GENEIOUS software, go to your GENEIOUS software, and then create a new folder. Create a new folder. So you name it Pairwise sequence alignment / mutation analysis. Mutation analysis. Click OK. So here is it. So this Pairwise sequence alignment and mutation analysis. So this is the folder you just created, and then you go back to file and import from file. So you are importing the sickle cell data, which is human HBB mutant gene, and then you alight both. So you click control and then alight, pick the second one and then import. So you keep sequence separated. Yeah, so we have imported data now. So this is the mutant hemoglobin beta gene, and this is the normal hemoglobin beta gene. So right now we'll be working with both sequences, and I told you something, that Pairwise sequence alignment is aligning just two sequences, only two sequences. So we'll be working with two sequences to run Pairwise alignment and then embark on mutation analysis. So just follow me very carefully so that you'll be able to understand better. So you click both, you alight both. So you click here, then you go to align and assemble. But before then, I just want you to see something. This is the hemoglobin normal gene now. As you can see, it's very, very long, because the total number of sequences here is about 73,308 nucleotide bases, while that of the normal, that of the mutant gene is about 1,577 nucleotide bases in the sequence. So as you can see, it's way smaller than in number than the normal gene. Are you getting it now? The normal gene now. So this is telling you that this mutant gene was extracted only at the region of mutation, where mutation take place in hemoglobin beta gene yielding to sickle cell. So the regions that were extracted for the mutant gene were the region where mutation took place. So all you need to do right now is go to align and assemble. So you pick Pairwise alignment. So Adebin is multiple sequence alignment. You're going to see multiple sequence alignment in this place, because that's the option the GENEIOUS software is going to provide, because it was already recognized that you're working with multiple sequence alignment. So but here the software recognized that we're working with this sequence, so we brought Pairwise sequence alignment. So you click and then you find this. So just like the procedure for multiple sequence alignment, so you click let's work with local alignment. Remember, this is alignment with free gaps. That means it's telling you to just align the area that is has high similarity, especially the areas that involved with mutation. So and you're going to have free gap, but this time around we're choosing local alignment, so there's no need for us to work on free gap. So it's just going to give us areas that are highly aligned with the mutant sequence and the the normal gene sequence. So and then trim both ends. So let's just run it. So you pick local alignment, and then we are working with 100% identical, and then you leave the rest and run. So you run. So it's going. So this is the result. As you can see, both ends are trimmed, and we have only about 1,577 sequences here. So that means it has been aligned. But this is for you to understand that immediately you are being given your result, you need to understand the nature of the alignment by looking at the consensus sequence. And this is the consensus sequence now. Why this is the graph? So look at the consensus sequence and look at the graph. Now this is the graph. So you see this area. This is for you to understand that there's an issue here. This for you to understand there's an issue. Remember I told you from here to here, as you can see, there's no issue, there's no alteration, there's nothing. So this is just to tell you that this area are highly aligned. From here to here, highly aligned. Only this area are not aligned, this area are not aligned, this area are aligned. So we're going to be looking and studying various issues that are related to these pairwise alignments when it comes to mutation analysis. So all you need to do is now is zoom. Yeah, so here we are. Now I already told you what alignment is all about, and alignment deals with seeing into the same nucleotide bases that a particular sequence share with another. So as you can see here, this area are aligned, and the graph here is making us understand that truly this area are aligned, and this is the consensus sequence. Now the consensus sequence is the result of these two sequences depending or being determined by the normal sequence. Are you getting what I'm saying now? Being determined by the normal sequence. So when we see consensus sequences like 1 + 1 here is equal to 2, so this is just telling us that if t is here, then definitely the consensus sequence is going to be t. So if t here, which is timing, t here timing, definitely the consensus sequence is going to be what? T. So the same thing with this one, CC, then consensus sequence is C, then AA, you understand, the consensus sequence is going to be A. So just like that. But whenever you find T here and you find G is here, then definitely there's an issue. The consensus sequence is not going to give you anything, but it's going to give you a portion of probability, trying to tell you maybe it's either A or it's either G or T that's going to be here. And you get what I'm saying now. So but you will understand what I'm trying to say as soon as we go. So this is just trying to tell you that from here to here they are all conserved, they are all aligned, because we have CC, this area is conserved, this area is conserved. That means they are all aligned, you understand. So you keep going, keep going. Yeah, this is an issue. Now for you to understand an issue, or for you to understand if a problem have arrived, or maybe you need to study a particular thing when it comes to mutation analysis in pairwise sequence alignment. So it is the graph that will tell you an issue, because normally you may not know if actually an issue occur between two sequences when it comes after the alignment now. But for you to understand if there's any issue, it is looking directly from the graph and the consensus sequence. As you can see, the consensus sequence is giving us Y instead of an aligned consensus in a nucleotide based being determined from the two sequences. But this one is telling us that is Y here, so which means it is giving you a probability of how can I put it now, a probability of maybe it is either C or it is either T, a probability of nucleotide sequence that might exist here. And then we have the graph, which is actually telling us a gap. There's a gap here, so it is telling you that there's an issue here that you need to study. As you can see here, we have mean pairwise identity over all pairwise in the column, which is green identify 100% identity, while green brown is at least 30, while red is showing below 30. So as you can see here, so but we're gonna look into understanding it. So we have the normal sequence, which is this, but we have the mutant sequence, which is this. This sequence is actually a woman beta globin gene. So now in the normal sequence we have C here, as you can see. So in this region actually we have C here, then we have C on the mutant gene. So this is actually telling us that this area doesn't have any issue, but this area has an issue. Why? Because we have C here and we have T on the mutant sequence. So that means a mutation has taken place here, and we understand that in mutations we have three types of mutations, which is deletion, substitution, and addition. Now in this area we understood that in the mutant sequence we have T, so this is telling us that T has been replaced instead of C in the mutant sequence. So originally it was supposed to be C that's supposed to exist here, because on the original sequence, which is the normal gene sequence, we have C here. We have C. As you can see, we have C. So that means C is supposed to be here in the mutant sequence for it to align. But since it is a mutant sequence, that means an alteration has taken place, and the alteration that takes place is actually this T. So that means T was substituted in the mutant sequence instead of what? G in the normal sequence. Are you getting it now? So T was substituted. So what kind of mutation took place here? It is called substitutional mutation, because T was substituted for what? For C in the mutant sequence. Are you getting it now? Now this Y is actually giving you an information. Now there is something called IUPAC code for genetic data. IUPAC code for genetic data. So IUPAC code for genetic data. Now now mind you, we have a gap here. So the reason why we have gap is because a mutation has taken place, and which is actually this mutation we have here. Are you getting it now? So but this Y donates a meaning, which is actually telling you that it's either any of these two nucleotide bases that exist here. But how do you understand this? How do you deduce this? How do you understand what the nucleotide bases are supposed to exist here? So there's something called IUPAC code for genetic data. So you go to your Google, go to Google, and then you browse IUPAC code for genetic or for nucleotide. Let's put it this way: nucleotide. So IUPAC code, and you go to image. So yeah, here is it. This particular one. You download this particular one. As you can see, you download it. So you right click and then you save image as. So it's saved to your desktop. Saved. Then another thing is you download table amino acid table. Yeah, so here is it. You search for reasonable ones. Download this. Yeah, so let's work with this. Save image as. So it's save. You save at you are true. You go back to your GENEIOUS software so that we can continue. Now remember I told you that this Y deduce the kinds of nucleotide are supposed to be on the consensus sequence, and this is a consensus sequence. Now the consensus sequence is supposed to be representative, nucleotide representative or representative of nucleotide base that that determines representative of the nucleotide sequence that comes as a result of the aligned nucleotide bases between two sequences. Now so but why is deducing maybe it is actually one of those two sequences that's supposed to be here. So all you need to do is go to to where you download your image, desktop. So keep going. So here is it. You open. So yeah, now no, we are not working with this. We're actually working with the IUPAC code for nucleotides. IUPAC code for nucleotides. IUPAC code for nucleotides. IUPAC code for nucleotides. So here is it. So you can reduce the size. So we have Y here. Now in the hyper code for nucleotide, we have different alphabets, as you can see here. Symbol, we have G, we have A, we have T, we have C. Now all these are just alphabet, are just symbols, but each symbol represents a nucleotide base. So G represents G, which is guanine nucleotide base. A is A, while G is G, C is C. We have R, which represents which means G or A. So that means if you find R here, it is either G or A that's supposed to be here. So then we have Y. Now we have Y here, and then in the hyper code we have Y, and Y donates what T or what or C. So that means what's supposed to exist here is what T or C, but mind you, in the normal sequence it is C. So that means what's supposed to exist here in the consensus sequence is what is C, because in Y we have what C, not T. The T that exists here is what is the mutant sequence that was mutated through substitutional replacement. Are you getting it now? So we have C here, so that was supposed to exist in this consensus sequence is what is C. So now another thing you need to understand in this mutation analysis is that each of these mutation that took place, you understand, actually have a resultant effect on the amino acids. Are you getting it now? But we're going to work on that, but let's first understood the mutation that took place in the nucleotide. We are done with that. Let's move on to the other mutations that took place. So keep going. As you can see, all these regions are highly aligned. We don't have any alterations or mutational symbol or sign there. So you just keep going, keep going, keep going. So you keep going, until you find something like what we just deal with in the previous one. So we have donating an issue, and we have S here on the consensus sequence, which is actually donating or signifying another issue. So now the first thing you have to do is go to the sequence, the normal sequence, which is this, as you can see, UOI, which is the normal hemoglobin gene. So you go down to where the alterations took place, and we have C here, and then in the mutant sequence we have what G. So that means it's substitutional mutation that took place here, which means G was substituted instead of what, instead of C. And we have S here, which is giving us a symbol of maybe it is either C or G that's supposed to be at the consensus sequence. Now mind you, whatever is at the consensus sequence is a representative of what, of the mutant sequence, aligning with what with the normal sequence, aligning with the mutant sequence. Are you getting it now? So that means we have C here, and then we have G in the mutant sequence. That means G is mutated by substitutions instead of what, instead of C in the normal sequence. So, and then we have a gap here, the graph. So all you need to do is just go to the image, look for S. So S is either G or C. So S is either G or C, and we have G here in the mutant sequence, but what's supposed to exist here is C, because in the normal sequence we have C. Are you getting it now? So that means C was substituted instead of what, G was substituted instead of C. So that's it, and then you move on to the next one. Understood the kind of mutation that took place. So substitution mutation, but what actually was mutated there was C, replacing with G. So now here we have K here, and we have a gap here, which is signifying there's a mutation here, and then you go to the normal sequence, we have G. So, and then in the mutant sequence we have T. That means T was substituted in the mutant sequence instead of what, in the G. And then and K signify what, you go to K, we have K, which is either G or T. So, and then in the normal sequence we have G. So that means what's supposed to be in this consensus sequence is what, is G. Are you getting it now? You understand now? So what was mutated was what, was G, and replaced with what, T. So we move on to the next one. So keep going. Keep going until we find another issue. So all these places are all conserved, all aligned, so there's no issue with all those regions. There's no issue. So whenever you see a gap at the graph, that means there's an issue, just like this. So we have Y here, and Y donates what? Stand for what? Means T or what? Or C. So that means in the normal sequence we have T, and in the motor sequence we have what? C. So that means C was replaced in the motor sequence instead of what? T in the normal sequence. So that means what's supposed to be here according to this IUPAC code is telling us that it's supposed to be what? T or C. So that means T is supposed to be here because of what? The normal sequence is actually T. So what mutation took place here is substitution mutation. So you keep going. Keep going. Aha. So as you can see, this region is definitely different from all the mutation analysis when it comes to nucleotide bases that we have been working on from the beginning. So as you can see here, we have G, and here are the normal sequence, we have close gap. So what does it mean here? It means there's an addition mutation. So addition mutation took place. That means G is actually deletion mutation. A deletion what mutation? Yeah. So this is not addition, this is actually a deletion. So because as you can see, we have minus here at the normal sequence, which is a symbol of deletion. Are you getting it now? So when two sequences that are being pairwise to understand the mutational process that took place. So for you to understand the deletion process, is just that the actual normal sequence we have a gap, while the mutant sequence we have a nucleotide base. So as you can see, it is a deletion mutation. So anywhere you are saying this, what took place is what? A deletion mutation. So something was deleted here. And as you can see on the graph, it shows the gap, which shows that there's a serious problem here. Now this N, what does N signify? Just go to the image, the IUPAC code of genetics, and you find N, and N means what? G or A or T or C. So it is actually telling us that G, A, C, T, G can actually exist here, the consensus sequence. So it is that any of those things can actually exist there, but this time around we have a deletion, so we really don't know the kind of the nucleotide sequence that was deleted, but actually something was deleted. Are you getting it now? Yeah, something was deleted. So now that's that on that, to understand the kind of mutations that took place. Now this is for us to now understand the effect of those mutations on each amino acids. You understand the effect of the mutation when it comes to the base on the codons, and then the effects of mutation in the codons affecting the amino acids. So yeah, so we just pass a nucleotide, a mutation here. So here, so now all you need to do now is click translation. You click translation, as you can see. So you click translation. So the additional sequences that were added are all amino acids sequences, that is protein sequences for each of the nucleotide base sequences. So we have a consensus sequence. So the consensus sequence has its own protein sequence, while the mutant sequence has its own protein sequence, while the normal sequence have its own protein sequence, as you can see. So consensus sequence is protein sequence, mutant sequence which is what protein sequence, while the normal sequence which is protein sequence. Are you getting it now? Now all this you are seeing, this star, they all signify stop codon. Are you getting it down? So let's wrap. If you wrap, you find a whole lot of them, a whole lot of dark spots, and in the dark spot we have something like star. Are you getting it now? Star here. So they all signified stop codon. So let's just try to unwrap Yeah, so we're actually working on this alteration. Yeah, here are you getting it now? So we have a stop codon here, as you can see. We have a stop codon here. Now don't forget I told you the sequence of the consensus sequence has its own protein sequence, predicted by the codons here. As you can see, TGC code for C, and this C at the bottom of your GENEIOUS application. You find something like cursor before column 501 in bracket original base 62,061 most over so and so. So you find column 32 and G, which is actually column 32 here. Yeah, this is column 32, and then we have what we call G, which is a residue of 11, and a G is trying to make custom. That is G, which is nucleotide base. Why C here is making to understand that this C, it is actually a symbol coding for cytosine and amino acid. Are you getting a protein product? That is in the consensus. You can see in consensus. So we also have TGC at where in AU7, which is the actually AU7615, which is the mutant sequence. So we have TGC, which code for C, and C is what cysteine, while TGC goes for C, and C is still what cysteine. The same thing with consensus sequence. Are you getting it now? So then we have this aspect, which is actually the region where the issue of alteration, mutation occur here. So but you get to understand that there are times when mutation occur, giving rise to different nucleotide sequence in the sequence, and leading to different codons. Actually, some of these mutations can actually yield what's it called now? They can yield effects on the protein product. Are you getting it? On the amino acid that is being produced because of the change in the nucleotide base. Are you getting it now? So all we just need to do is try to understand what actually go on here. So now we have in the mutant sequence we have ATC. But before we do this, let me just show you. I told you that this is a stop codon. We have TGA. So how do you understand that that's a stop codon? You just need to go to the other sequence, which is this, and then we have what do we have there? TGA. So TGA is, and whenever we are saying G, because timing will change to U in RNA. So most times this code, this table are always shown in RNA form. So whenever you are saying U, it is actually T, and whenever you see T here, it is actually U on the table. So we have what? TGA as a stop codon because of this arch. So let's look at stop codon TGA. So we have TGA. So U is changed to T now. So that means UGA, which is actually TGA. Are you getting now? Which is stop codon. Are you seeing it now? It's a stop codon. It's donated by what? By ash. Now this ash here. So we have let's go to the normal sequence. So we have ACC. So that means ACC code for what? T, and T is what? Leucine, threonine. Yeah, T is threonine. So and that is, we have what? ACC. So let's look up for ACC. But first you look for threonine, TH. Yeah, here's it. And we have ACC, so that means that will be ACC. So we have it ACC, which is what? Threonine, as you can see. So that means the normal sequence ACC code for what? Threonine. Are you getting it now? Threonine. Actually this one, threonine. Are you getting it now? So that is the normal sequence. So let's now look at the effects on the mutant sequence. We have ATC. It is we have substitutional, mutation here that changes C towards T mutant sequence. So we are looking into understanding the effect of the amino acid that has been changed here. So we have ATT. Now ATT is what? ATT, just try to put it here and look at the bottom line of your sequence. Just put it on the here, as you can see. Then we have isoleucine. Now look for isoleucine on the table. Isoleucine. Coming. What actually the symbol? The symbol is ILE, which is this. So and then it's being coded by those codons, and we have ATC, that means AUC. Yeah, here's it. Isoleucine, AUC. Are you getting it now? So that's the effect of the what's it called now? The effect of the mutation in the codon yielding to this kind of amino acid. So that means the normal amino acid supposed to exist in the sequence. In the mutant sequence was supposed to be T, but because it's mutated, we have what isoleucine instead of what the threonine. Are you getting that? Now, so looking at the consensus sequence, like the protein sequence of the consensus sequence, we have T or I. So this is trying to tell you that what's supposed to exist here because of the probability of the Y, when it comes to the base. So that means what's supposed to exist here is what? T or Y, but because there's a mutation here, so it's giving us probability of T or Y or I. Are you getting a T or I? Are you getting it now? So now, how do you understand the very functional effect of this mutation? So all you just need to do is go to Google. So once you go to Google, click or you type threonine. You understand? Or you say the function. Let's put it this way. Function of threonine. So we have threonine is an essential amino acid, that is, it is vital for your health. Amino acid now, but it cannot be synthesized by your body and therefore has to be obtained from a diet. This amino acid supports central nervous, contravascular liver, amino system functioning, just to a new few. Are you getting it now? So this amino acid supports central nervous system, contravascular liver, immune system. So as you can see, the effects of this, the central nervous system, that is the functional result of threonine in the the cell, the sequence now. So we have cardiovascular, liver, immune system. Now let's talk about immune system, because actually the the sickle cell we are talking about is actually associated with immune system. So it is used or function for immune system function. And are you getting now? So another way is that the aspect of the nervous system. So there can be certain messages that have been transmitted from the hemoglobin beta gene to the brain for let's say involuntary actions that used to take place. So but because that is the normal function of threonine in the mutant sequence, as you can see this now, but because we have a mutational effect here on the mutant sequence, which has a change to isoleucine. So let's now look at the resultant effect of this. That means the function of threonine was replaced for the function of isoleucine here. You understand? Which result to the the effect of sickle cell in the human cell. So let's just look for the function of isoleucine that was replaced. So isoleucine. So it is particularly recommended to professional athletes and bodybuilders, as the primary function of isoleucine in the body is to boost up energy. And you understand it now? Energy levels. So assist the body in recovering from strenuous physical activity. So the main function of isoleucine is what? Energy. So this is trying to tell us that the function of threonine was replaced for with what? With a function of isoleucine, which is actually energy. Now the essence of this is that it improve or give more energy to the mutant sequence to stand against certain maybe effects of maybe CRISPR-R, which is, and CRISPR-R is actually a function. You can just make a search on that to make a resist or repair certain mutated areas in the in the nucleotide sequence of genes. Are you getting it now in the body? So but the energy given to this aspect is actually empowering this mutant sequence to stand against invasion of the response, the body response of the body against this mutation. Are you getting it now? So that's why it is associated with immune. You understand? So let's look at the other mutation. Let's look at the other mutation that took place. So let's just take it because of our time. Yep, so we have it here, and then as you can see this, we have CGC. Are you getting now? We have what? CPC. So now look at it. GCT code for what? A, and A is alanine. Why? CCT code for what? Proline. So the actual nucleotide, amino acid that's supposed to exist here is proline, but because the mutation has took place here, we have what? Alanine. So let's try to check that again. The function of proline. Yeah, proline plays important roles in protein synthesis and structure. Protein synthesis and what? Structure. Metabolism, particularly the synthesis of adenine, polyamine, amides, and glutamate via, and nutrition, as well as wound healing. Imagine wound healing. So the essence of this proline here in the normal sequence is for maybe healing. Maybe when there's an alteration somewhere, it's supposed to function for what? For healing. Are you getting it now? For healing. Are you getting it now? And protein synthesis, that's it. Synthesis of certain proteinous functions in this gene. So but then because we have a mutation that took place here, it give us alanine. So let's say the function of alanine, so that means the function of proline replaced with alanine, which is alanine is an amino acid that is used to make proteins. Tryptophan and vitamin B is a source of energy for muscle and the central nervous system. It strengthens the immune system, so it's still a kind of form of energy too. Are you getting that? So that is actually associated with this. So the function of proline was replaced with alanine. You understand? Replace with alanine. Are you getting it now? It was replaced with alanine. So now this actually consistency are just telling me it's either protein or alanine, but we understand that what's supposed to be here was what is what? Proline, but replaced with what? Alanine here. So that's why there's alteration here. So let's just keep going. So we have another one here, so you can deduce this. Yeah, we have arginine being replaced with what? Isoleucine, the function of arginine. So just try to do that yourself. Yeah, just try to do that yourself. So let's keep going. But there are times that we have a change in codons, in nucleotide base that yields a particular codon. Are you getting that? It yields a particular codon. Yeah, and then the codon code for an amino acid, but the alteration that took place, or the mutation that took place, did not actually change the effects of protein or the protein product. This amino acid that's supposed to code for according to that codon. Are you getting what I'm saying now? So what I'm trying to say is that we have a mutation that took place in let's say in this sickle cell now. So there are times that the mutation that took place in the bases did not actually affect the amino acid. That's what I'm trying to say. Mutation take place but did not affect the amino acid. Yeah. So this is it. TCT, YTTT. Actually, a mutation took place here, but the mutation that took place did not change the amino acid. So this is a silent mutation. We have silent mutation, we have nonsense mutation, and we have missense mutations. So you can just search that on Google. I repeat, search it on Google and try to understand. But what took place here is just a silent mutation. Why? Because it did not affect the amino acids. Are you getting it now? It didn't. And then nonsense mutation, missense mutation actually affects. No, it does. Missense actually affect the amino acid. So you can just search that on Google and you have deep understanding on it. So the change in this isoleucine, so it really doesn't affect the gene. So now let's go back to our deletion mutation. Yeah, where is it? Yeah, here's it. Now in this deletion mutation, actually there are a lot of things here. So this is X, and X symbolize unknown. Are you getting it now? Unknown. Why N also symbolize unknown? Why X symbolize unknown? Are you getting it now? So if you look here from this gap, the codons are supposed to form here is from here to here for G. Are you getting now? And G is what? Glycine. But because there is a deletion mutation, so this will tell us that something was deleted here. You understand? And it actually affects the protein structure. It affects the protein structure. So this is GGG, which is actually the mutation. You understand? But the whole of this from here to here, affect the protein structure, giving rise to an unknown, functional proteins. You understand? In the body, which really alter the functions of hemoglobin beta gene, yielding, resulting to sickle cell, disease in the body. So I think we've come to the end of pairwise and mutation analysis. So we're going to work on another mutation analysis on the next curriculum. So please do your assignments on an area using pairwise alignment, which is, that means you run pairwise alignment of the normal gene and the mutant gene of pairwise alignment and submit to the email so that I will go through it and get back to you. Do that. So thank you. Have a nice time.